Hello everyone. In this video, we'll talk about a paper entitled Towards a Consistent Evaluation of microRNA Disease Association Prediction Models. What is microRNA? It is a non-coding RNA with length of around 22 nucleotides. It plays a role in regulation of gene expression. Thus, microRNA is a promising drug target. Researchers utilized machine learning, specifically the supervised machine learning method, to identify associations between microRNA and disease. In this paper, Dong and Hosla studied 3 prediction models for microRNA disease association, identified 3 common issues in the prediction models, and provided solutions, as well as implemented ablation studies to observe the performance of the suggested variants from the 3 prediction models. Now, let's see the similarity measures of microRNA and disease used in the prediction models. There are 4 similarity measures, which are disease semantic similarity, microRNA functional similarity, microRNA sequence similarity, and Gaussian interaction profile GIP kernel similarity. These similarity measures are the input for the prediction models. Next, let's look at the 3 microRNA disease association prediction models. First, the age perturbation-based method for microRNA disease association prediction (EPDMA) that extracts from microRNA disease bipartite network, microRNA similarity network, and disease similarity network is trained using the 5-layer multi-layer perception regressor to obtain the output. This study suggests a variant EPDMA1 where the MLP regressor is replaced with linear regressor. The second model is the neural inductive matrix completion with graph convoluted network NIMGCN. Each input, the disease semantic similarity and microRNA functional similarity, used 2 graph convoluted networks GCN encoders to learn the node representations, and multiple neural projection layers is applied to extract the output. There are 3 variants explained in this study, which are NIMGCN1 that removes the 2 GCN, NIMGCN2 removes the neural projection layers, and NIMGCN3 that use only 1 GCN and 1 NPL. The third model, DBMDA architecture, is where both disease semantic similarity and microRNA functional similarity becomes the input for one autoencoder, and the microRNA sequence similarity is the input for another autoencoder. The extract from these autoencoders is then applied to a rotation forest, which will produce the output. The variants DBDMA1 remove both autoencoder, DBDMA2 remove the microRNA sequence similarity, and DBDMA3 remove the microRNA sequence similarity as well as the autoencoders. Now, let's move on to the next part, the major problems for current models.
Data output drawback is one of the major problems for the current machine learning models. There are 2 data sets, so-called training set and test sets, are used for constructing models. During model training, the parameters are learned from the training set, whereas the evaluation of the model competency to predict the unreviewed data is dependent on the test sets. Data outflow happens when both sets are trained in the models. In lieu of processing only the training set, the additions of the test set cause the prediction ability of the model to be exaggerated. This will eventually lead to the biased comparisons, especially for the model which utilizes miRNA functional similarity or GIP kernel similarity, due to these evaluation similarities being pre-computed. To hinder the bias, the pre-enumerated similarities are included in the training set only during predictive evaluations.
Second, the evaluation setup. The models use random seeds, and they use different training and testing set as well as the evaluation matrix. These factors will affect the fairness in comparing the models.
Furthermore, the last main problem of the current model is regarding the building process of models. Overfitting can occur when building more complicated models, although the model performance may be excellent. it cannot be denied that the more complex a model is built, the more training sets are required to accommodate the surge in parameter use. Therefore, ablation studies are highly demanded to generate complex models for predicting the associations between mRNA with its related disease. For data gathering protocol, the datasets were obtained from HMDD2 and HMDD3 database. The amount of miRNA and diseases found in the HMDD2 and HMDD3 database and their known correlations were tabulated in Table 1. MESH was compared to rule out the missing diseases that were not in HMDD2 and HMDD3 database. Then the data were tabulated in Table 1. The missing diseases were used for similarity matrices based on GIP-KQUEEN similarities. Moreover, for setting out protocols of known and unknown mRNA disease correlation, data from known mRNA and disease correlation were split fivefold, k equal to 5, to test and train the data. This method was known as fivefold CB. After 5 times of fivefold CB, the data were trained and tested with random seed to generate the same training and test sets in the 3 models. After 25 times training, 25 training and test sets were generated and split. The performance score AU, ROC, and AP were calculated after generating the same training and test sets among the 3 models. Another performance scoring, top K, was computed for the top 5 framed cancers. Then, the procedure was repeated by splitting data from unknown mRNA disease correlation to form balanced and unbalanced datasets. Furthermore, for protocols of input resemblance training in models, the input resemblance of each models were enumerated as Table 1. The red color indicates the input resemblance calculation was repeated 5 times by using heterogeneous inputs. For the last part of the methodologies is the building of the hyperparameters. An autoencoder was utilized for DBMDA hyperparameter training. Encoder and decoder were the 2 components which made up an autoencoder. For NIMGCN and EPMDA models, the hyperparameter was computed according to literatures. The encoder was linked to a 32 size layer, while the decoder was imputed with identical size. Then, the autoencoder was trained for 100 reports. Then finally, the parameters were recommended to remain default for the rotation forest classifiers. The first part of the result is regarding the data gathering, in which the data are collected from 2 databases, HMDD2 and HMDD3. The amount of mRNA and diseases that can be found from both databases are tabulated as Table 2. The known correlation between mRNA and its related diseases are then calculated from the both databases. However, there are some diseases missing when comparing both databases with MESH. Therefore, the missing diseases are then used for filling and computing into resemblance matrix. For the third part of the result consists of scoring of AU, ROC, and AP for the 3 models based on HMDD2 database. The model which use pre-enumerated resemblance are displayed as erroneous result, while the model which implements training sets that are computed during the experiment are shown as correct results. From the graph 1 shown, the erroneous result display greater scoring when compared with the correct results. So, the model EPMDA has the largest differences in performance scoring. Due to this model, it depends on GIP-KQUEEN similarities. The result proved that the use of disease thematic similarities in the models, such as NI, MGCN, and DBMDA can maintain the performance levels. Moreover, the additions of mRNA sequence similarities and DBMDA models can protect the model from the decline in the performance. Moreover, for the fourth part of the result is shown in the graph 2. There is one model, DBMDA, and its variants are not included in the graph 2 due to the extraordinarily low performance levels. Observing the data from the NIMGCN models, the imbalanced set of the training mildly surged the performance scoring in the HMDD2. Nevertheless, the implementation of imbalanced training data reduces the performance level in the HMDD3. For EPMDA models, the performance levels of balanced and imbalanced training data contrast with the EPMDA1 for the both HMDD database. Overall, the higher performance scoring in HMDD2 database is EPMDA1 for balanced training sets, while the greatest performance scoring in HMDD3 database is NIMGCN3. Therefore, a balanced training set is suggested to be used in the models for better performance. Lastly, for the mean score of AP among the three models and their variants are computed on both databases by using balanced training sets. The AP scoring is one of the performance evaluation metrics. By viewing the graph 3, the lighted models are having lower standard deviation compared to original models. From the overall result displayed in graph 3, EPMDA1 and NIMGCN3 possesses the greatest AP score for HMDD2 and HMDD3 database respectively. In conclusion, the current machine learning models possess some degree of problems. For instance, some current models have data output problems, evaluation setting problems, and model building problems. Therefore, this article recommends some suggestions to improve the current models by computing input resemblance from the training sets, utilizing balanced training datasets, imputing negative testing samples, and using AP scoring as the performance evaluation metrics. Besides that, these articles also emphasize on the need of the ablation studies for generating more complex models with additional new features.